Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tool, that is restriction enzyme, polymerase enzyme, ligase, vector, and the host organism. Let us try to understand some of these in detail. Restriction enzyme. In the year 1963, the two enzymes responsible for the restricting the growth of bacteriophage in Escherichia coli, known as E. coli, were isolated. One of these added methyl group to DNA, while the other cut off DNA. The latter was called restriction endonuclease. Paul start. The first restriction endonuclease हिंद टू, इसका फुल फॉर्म होता है हीमोफिलस, मतलब इसका उसके बाद ही हो गया मतलब रेट functioning depend on the specific DNA nucleotide sequence was isolated and the characterized 5 years later. It was found the Hind to always cut DNA molecule at a particular point by recording a specific sequence of the 6 base pair. This specific base sequence is known as the 6 base pair. This specific base sequence is known as the recogenating sequence for Hind2. Basis Hind2, today we know more than 900 restriction enzymes that have been isolated from the over 20 to 30 strain of bacteria. Each of which recording different recogenation sequence. The conversion of naming this enzyme is the first letter of the name come from the genus and the second two letter कहाँ एक मिनट लो second two letter comes from the species of prokaryotic cell from which they were isolated. The EcoR1 comes from the Escherichia coli. In EcoR1, the letter R is derived from the name of strain. Roman number following in the name of indicate the order in enzyme belong to the large class of enzyme called nuclease. These are of two kinds: exonuclease. Exonuclease remove nucleotide from the end of DNA, whereas endonuclease may cut at a specific position within the DNA. Each restriction endonuclease function by inspecting the length of a DNA sequence. One it find its specific recombination sequence. It will bind to the DNA and cut each of the two strands of the double helix at a specific point in their sugar phosphate backbone figure. Each restriction endonuclease reconnaissance a specific palindromic nucleotide sequence in the DNA. इस डायग्राम को देखिए और समझिए. Thank you for watching.